In this video, we learn about animal nervous systems. All of the movements in an animal body are triggered by electrical signals that are communicated by nerve cells called neurons to muscle cells. Everything that we do, including moving, seeing, and thinking, are based on the flow of ions across the membrane of cells that are part of the nervous system. All animals except sponges have neurons and muscles that are involved in these kinds of responses. The neuron transmits the signal, and the muscle launches a response to the signal that is received by the body. The cells that are responsible for receiving information from the environment are called sensory cells. These cells respond to various stimuli, including light, sound, touch, etc. The sensory cells present in different parts of the body, including the skin, eyes, ears, and nose, transmit streams of data about the external environment. The sensory cells that are present inside an organism's body are responsible for monitoring conditions that are important in maintaining homeostasis. For example, maintaining blood pH, oxygen levels, and controlling the rate of the heart. These cells also transmit the information they receive from the environment to the brain and spinal cord through nerve cells known as sensory neurons. The incoming sensory information is integrated in the brain and spinal cord, which make up the central nervous system, or CNS. Clusters of sensory neurons bundle together into long tough strands of nervous tissue, forming structures called nerves that transmit sensory information to the brain and spinal cord. Another kind of neurons, called interneurons, that are also part of the CNS, make connections between sensory and motor neurons. Motor neurons are nerve cells that send signals to effector or response cells in glands or muscles. All of the components that are present outside of the central nervous system form part of the peripheral nervous system, or PNS. If we look at the human body, we take in a lot of information from our surroundings through sensory cells that are present, for example, within eyes. Sensory cells within the eyes transmit this information to the brain and spinal cord via sensory neurons present in bundles called nerves. The brain and spinal cord, which form the CNS, then process this information and send commands down via interneurons to motor neurons, which signal the muscle tissue to launch a response to the incoming signal. This response may include, for example, the action of turning pages in a book. If you look more closely at a neuron, it has three distinct parts. The first part consists of dendrites that bring in information from surrounding neurons. This information then passes through the cell body that is involved in processing the signal and generating an outgoing electrical signal, which then travels down through the axon to the axon terminals and subsequently to other neurons that the axon terminals connect to. And subsequently to the dendrites of other neurons that the axon terminals connect to. Cells, especially those that are part of the nervous system, are electric in nature meaning that they have the ability to conduct electricity because of the unequal distribution of ions on both sides of the plasma membrane, which creates an electrical potential. When this potential exists on either side of a plasma membrane through the separation of charges, it is called a membrane potential. These potentials are measured in units called millivolts. There are generally more negative ions present inside of a cell than the outside, and since the membrane potential is measured depending upon the concentration of ions inside relative to the outside of the cell, we generally use a negative sign when referring to this membrane potential. In neurons, membrane potentials are typically between negative 65 and negative 80 millivolts. When a membrane potential exists, the ions on both sides of the membrane have potential energy. If this membrane was removed, these ions would spontaneously move from an area of like charge to an area of unlike charge, leading to a flow of charge in the form of an electric current. But when you do have a membrane separating charges, ions move in response to their concentration gradients as well as charge gradients across that membrane. This combination is called an electrochemical gradient. Now, ions being charged cannot easily cross a lipid bilayer by themselves. Since they're polar or hydrophilic, they need the help of a transport protein or an ion channel embedded within a plasma membrane to allow the flow of charged ions into and out of the cell. At rest, the voltage of a neuron is called the resting potential. The reason that this resting potential exists is because there is a higher concentration of positive charges outside of the cell relative to the inside. The main cation inside cells is potassium, and the main cation present outside the cell is sodium. In addition to sodium, chloride ions are the main anions present outside cells. Inside cells, the major anions are protein molecules that have an overall net negative charge. If we used an instrument that measures the voltage across membranes, we would find that it would be a negative 65 millivolts. This is because of the separation of positively and negatively charged ions and molecules across the membrane of a cell. In neurons, the main positively charged ions present outside of the cell are sodium ions, and the main negatively charged ions present outside the cell are chloride ions. Inside the neuron, the main positively charged ions present are potassium ions, and the main negatively charged molecules are protein molecules. So the charge across the membrane depends upon two factors. One is the concentration of ions on both sides of the membrane, and second, the charge distribution across the membrane. Since the plasma membrane is selectively permeable, only certain substances can cross it. So these ions can only cross plasma membranes efficiently in one of three ways. One, along their electrochemical gradient through an ion channel. Two, carried via a membrane cotransporter protein or an antiporter protein. These proteins are able to move two ions at a time in the same or opposite directions along a plasma membrane. And three, ions can be pumped against an electrochemical gradient by a membrane protein that hydrolyzes ATP, since it takes energy to move ions against their electrochemical gradients. A neuron at rest is able to maintain its resting potential because of the movement of potassium ions across the membrane and out of the cell. Resting neurons are most permeable to potassium ions, which cross the membrane easily along their concentration gradient. Potassium ions move through channels within the membrane called potassium leak channels, because they allow potassium to leak out of the cell. There are many potassium leak channels in the membrane of a neuron. As potassium moves out of the cell, the inside becomes more negatively charged relative to the outside. This contributes to the resting membrane potential of a neuron. The movement of potassium ions through the potassium leak channels allows the membrane to reach a voltage where there is equilibrium between the concentration gradient that moves potassium out of the cell and the electrical gradient that moves potassium inside, and this is called the equilibrium potential for potassium. Chloride and sodium ions also cross the plasma membrane, but much less readily than potassium because there are fewer chloride and sodium ion channels within the neuronal membrane. But these ions also have their own equilibrium potentials. Another factor that contributes to the resting membrane potential in a neuron is the sodium-potassium pump, also known as the sodium-potassium ATPase. This pump is responsible for establishing the resting membrane potential. It imports 2 potassium ions and exports 3 sodium ions, resulting in the concentration of potassium ions being higher inside and sodium being higher outside. Thus, the inside of the neuron becomes more negatively charged with respect to the extracellular environment. Because although potassium ions are flowing into the cell, the electrical charge inside the cell is still dominated by the overall negative charge of protein molecules, which are present inside the cell in high concentrations. As a result, the neuron has a negative resting potential. This image shows how the sodium-potassium pump works. First, 3 sodium ions enter the pump on the inside. Then, using energy from ATP, the pump changes shape, transporting these sodium ions out of the cell. This is followed by the binding of 2 potassium ions on the outside of the cell. So the pump picks up 2 potassium ions from the outside and then changes shape again to release these ions inside the cell. Thus, more positive charges are being pumped out than in, resulting in a net positive charge outside the cell and a net negative charge inside the cell, leading to a negative membrane potential in a neuron at rest. This brings us to the end of the section on neurons and how they maintain their resting membrane potentials. At the end of this section, I would like you to be able to answer the questions listed on this slide. Thank you.